Hello everyone, today's lesson is mitosis. Now mitosis basically describes the division of the nucleus, and that's what we're going to talk about today. So every hour we have tons of cells dying off, but they're also replaced at an equal rate. And this is through a process called cell division or mitosis. There are a few reasons cells divide. The first is growth. So as we grow, we need to increase the number of cells. So we go from being a tiny wee baby into a mature adult, and to get from point A to point B, we need more cells because we are physically growing in body mass. So the first reason is to grow. The other reason is for reproduction. Now, when we're talking about mitosis, mitosis is how non-reproductive cells divide. We also call them somatic cells. So somatic cells are found in things like skin, muscle, lungs, gut, things that aren't involved in the process of reproduction. When we talk about cells dividing for the purpose of reproduction, we're largely talking about the process of meiosis. Not mitosis, but meiosis. So that's a concept that you will look at if you go into higher levels of biology. Today we're focusing on mitosis. The final reason that cells divide is to repair. And repairing helps to replace any dead or damaged cells. Remember, our two different types of cell death, necrosis and apoptosis, so either damaged or old cells. And we also replace cells that have a limited life. So basically just to repair cells that aren't functional anymore. In our last lesson, I mentioned IPMAT, where I stands for interphase, P is prophase, M is metaphase, A is anaphase, T is telophase. And so PMAT is the process of mitosis, where the cell nucleus is dividing. And interphase is that phase before mitosis where it's preparing for mitosis. What you will need to have to do is be able to sort of describe what is going on in each phase, as well as be able to correctly identify the phase if given like a microscopic image or a photograph. So let's get into that. Interphase, we can think of it as an increase. So an increase in organelles and in DNA, because we're doubling the DNA and we're increasing all the organelles, preparing to be split apart. Prophase, we can think of as prepare, because a few things are happening in prophase that is preparing to allow us to split. In metaphase, things are moving to the middle. In anaphase, they're pulling apart to two sides, and in telophase, we end up with two nuclei. And I'll get into more detail, but prepare, middle, apart, two, or PMAT. We talked about interphase quite a bit in our last lesson, but basically it's the cell preparing for cell division. And it does this by growing, making more organelles, and copying its DNA. We know that there are three phases: the first growth, the synthesis, and the second growth. If we look at a picture here, you can tell that it's interphase because you can clearly see the nucleus is still intact, and it looks sort of like a normal cell otherwise, but you can tell that it's an interphase. Okay, so we get into the P of IPMAT, or prophase, which is also prepare. So what happens in prophase is the DNA condenses, and it does this into, it condenses into chromosomes, and we call them, like another way of saying it is sister chromatids. The other thing that happens to prepare is that the nuclear membrane starts to dissolve. So it breaks apart, and this sort of releases the chromosomes out into the space. And it looks like my slide got a little bit messed up here, but what it says is the centrioles move to opposite poles of the cell. So that just means that they're going to be hanging out on the opposite ends of the cell. You'll remember that the centrioles were created in our second growth phase of interphase. If we look at these pictures, you'll see a picture of early prophase and late prophase. You can tell that it's prophase because we have these sort of chunky things, and that's actually the chromosomes that you're seeing. The difference is that here our nucleus is maybe sort of still intact. We get that dark red splotch in the middle, whereas down here in late prophase, our nucleus is totally come apart and the chromosomes are fully formed. Here we have chromosomes that are sort of starting to come together into those distinct shapes, but not quite there yet. Metaphase. Remember, the M is for middle. So the M is for middle, and if you look at our images, those chromosomes, those condensed chromosomes, are lined up in the middle of the cell. So that's what metaphase stands for. So the chromosomes get to the middle because spindle fibers attach at their centromere. So if we look at a chromosome, the centromere is at the center. Okay, so we have spindle fibers attaching to the centromere, and then it pulls the chromosomes into the middle of the cell, and we also call this the equatorial plate. The middle of the cell is the sort of equator of the cell, the equatorial plate. Anaphase is the next step. So anaphase, A, stands for apart. So first we lined them up in the middle, and now we're pulling them apart because remember, we need to divide these chromosomes so each daughter cell will get a full copy of the DNA or genetic material. If we look at the images, you can see there's the boundaries of the cell, and the chromosomes are clearly being separated into two different poles. Here you can actually see the spindle fibers pulling them apart. On this slide, we're going to talk about telophase, which ends mitosis, and cytokinesis. Telophase is basically where a nuclear membrane forms around two groups of sister chromatids. Okay, so that's telophase, and that's the end of mitosis, because you'll notice we now have our new nucleus formed because the membrane has formed around our group of sister chromatids. The other very important process involved in cell division is that called cytokinesis. Cytokinesis refers to the division of cytoplasm. So telophase is only the nucleus. Cytokinesis is the cytoplasm and the rest of the things, the organelles in the cytoplasm being divided. So how this happens is the cell membrane forms what's called a cleavage furrow. And what that sort of is basically a sort of pinching of the membranes. And so you'll see this little line when cytokinesis is occurring. You'll notice it says cell plate. That's because technically these are plant cells and we call them something different. I'll get into that in a minute. So cytokinesis is that cleavage furrow forming and splitting the cytoplasm and all the organelles into two unique cells, whereas telophase is the formation of a nuclear membrane around those two groups of sister chromatids, creating a new nucleus. So the end result of mitosis and cytokinesis is we get 2 new daughter cells. These daughter cells are genetically identical to the parent cell, so they are the exact same, but we call them the 2 daughter cells. So we don't say that, the old cell and the new cell. That's not how we refer to them. It's daughter cells. So here is a cartoon sort of depiction of the process. In interphase, we have our organelles growing and duplicating. We have our centrioles appearing, and just generally getting ready for the rest of mitosis. In prophase, our centrioles are moving to opposite poles. Our DNA is condensing into chromosomes, and we're developing spindle fibers in order to pull those apart. Additionally, our nuclear envelope is breaking down, so here you see it in little fragmented pieces. During metaphase, or middle, our chromosomes are lined up along that equatorial plate. Anaphase, the sister chromatids are being pulled apart, and they will be genetically identical to each other. So these two are the exact same, these two are the exact same, these two are the exact same, so that each new cell gets a complete set of DNA. In telophase, we have our nuclear membrane reforming and our cleavage furrow, which is the process of cytokinesis, where the cytoplasm is being split into 2 cells. If you're in my class, take a moment to go watch one or both of these videos. I think one of them is Crash Course and the other might be the Amoeba Sisters. They're great videos that talk about mitosis. Here's where you have to be a bit careful. When we're talking about genetic material or DNA, there's a lot of C words that we use to describe it. So, chromatin is basically DNA wrapped up in proteins that help it to sort of stay together in some sort of form. Here we have uncondensed DNA, and for our purposes, we can just call it chromatin. When the genetic material condenses, we get 1 chromosome. When it duplicates, we still have 1 chromosome, or 2 sister chromatids, and that says that right there, 2 sister chromatids. So this is a sister and this is a sister chromatid, or this is a sister chromatid and this is a sister chromatid. But this whole thing is known as a chromosome. So when these sister chromatids are connected with this centromere, then the sister chromatids together are a chromosome. Once they're split up, then we actually have 2 chromosomes. So it's definitely a bit confusing. Now, when we're talking about differences in mitosis between plants and animals, in animals we have that cleavage furrow and happy day 2 daughter cells, easy as that. In plants, it's a little bit more complicated because of that cell wall. We instead have basically vesicles that move to the center of the cell and form what's called a cell plate. Then the existing cell wall will connect with that cell plate, and then we get the 2 daughter cells. So the biggest difference is that in plants we have this cell plate forming, whereas in animals we have that cleavage furrow. So, last concept. Your cells are not constantly dividing. Remember that cell cycle arrest phase, or GO? Different cells are going to experience mitosis, or a rate of mitosis, differently. And it depends on what the function of the cell is. For example, onion root tips is where growth of the plant occurs. So these root tips are stretching out, they're trying to acquire sort of more resources in the ground, and that's where the growth is primarily occurring. And so those cells will divide very frequently. This is in contrast maybe to other parts of the onion that aren't actively trying to grow, and so they will experience mitosis at a much slower rate. The same idea goes for, animals. If you recall, one of the purposes of mitosis is to repair damaged cells. Now, our skin cells or epithelial cells are constantly being damaged by our environment and so have to be replaced constantly. So what happens is you'll see skin cells on the top layer or superficial layer of the skin being damaged, and they'll fall off, and that's okay. They're dead, whatever. And what happens is the cells underneath will be dividing in order to sort of ensure that our body is still protected by a sufficient layer of cells. So that's the lesson on mitosis today, or cell division.